infect bacterial cells. Bacteriophages have been the most extensively studied bacteriophages are polyphages. Polyphages are those viruses who have the capacity to infect coliform bacteria. Now, amongst these polyphages, there are T1 phages, T-odd phages. T1 phages or T-odd phages, they are the phages, polyphage, they are belonging to the polyphage group. These polyphages make the most extensively studied multiplication cycle of the bacteriophage is T4 bacteriophage. In that T4 bacteriophage, which is the example of virulent phage or lytic phage. T4 bacteriophage is the example of virulent phage or lytic phage. In this T4 bacteriophage, the way of multiplication or the way of replication is mainly by the lytic cycle. Now, lytic cycle means what happens? In the lytic cycle, the virus genetic material will be replicated and it will be followed by transcription and translation. The proteins are synthesized. The lysis of the virus or killing of the lysis of the bacterial cell will be carried out. In case of another type of replication which is seen in the avirulent phages or that is seen in the temperate phage, that cycle of replication is called lysogenic cycle of replication. Now here in this lysogenic cycle of replication, the genetic material of the phage, that is called phage genome, attaches, followed by penetration in the host cell, and after penetration in the host cell, here the phage genetic material, instead of undergoing lytic cycle of replication, what happened? That phage genetic material from the lambda phage has been incorporated in the host genetic material, and because of incorporation of that phage genetic material in the host genetic material, the killing of the host cell or lysis of the host cell will be prevented. In the first generation, the viral genetic material will be incorporated in a bacterial genetic material. That bacterial cell is called a prophage, in which the phage genome has been integrated in a bacterial genetic material. That prophage undergoes multiplication. Now, please try to pay attention. See, here in case of bacterial cell, what happens? Bacterial cell undergoes multiplication by transverse binary fission. Bacterial cell undergoes multiplication by transverse binary fission, which is one mode of multiplication. In a bacterial cell, transverse cross walls are formed. Equal distribution of all the components will be carried out in both the compartments, and then cell divides. Here, in case of bacterial cell in whom prophage DNA is there, it will undergo also replication, it will also undergo multiplication. The prophage has been observed in both the cells, daughter cells, in both the daughter cells. Now, when you will take into consideration a prophage, that prophage is called lysogeny, in which the viral genetic material is present in the bacterial cell. Now, that bacterium in whom the phages will undergo lysogenic cycle of replication, that is called temperate phage or avirulent phage. it undergoes a lysogenic cycle of replication that is called lysogeny. In case of a specific type of proteins, please try to pay attention. Here, when the bacterial cell में phage genetic material has been incorporated, a specific type of immediate early proteins will be synthesized, and because of synthesis of that immediate early proteins, what will happen? कि normally the replication followed by transcription and translation takes place. Phage का independently, bacterial cell का independently, and lytic cycle will be operated. On the other hand, here in case of lysogen or prophage, what happens? The genes which are responsible for synthesis of immediate early proteins, that genes are suppressed or repressed, and the genes which are responsible for integration of viral genetic material into the host genetic material, that is progenes, they have been activated. As a result, suppression of lytic cycle will be carried out, and the activation of lysogeny cycle will take place. Here, the prophage DNA will be formed, and that prophage DNA will be carried out for a series of generations. Now, what happens in case of bacterial cells in whom the lysogenic cycle is there? The viral genetic material is present in the bacterial genetic material. A segment of viral genetic material is present in the bacterial genetic material, and because of that segment of viral genetic material which is incorporated in the bacterial genetic material, the phenotypic characteristics of the host are changed. Now, the most important phenotypic characteristics of the host which are concerned with the virulent activity, which are concerned with the antigenic properties, which are concerned with the toxin productions, they are associated with the phage, they are associated with the prophage, and when they are associated with the prophage, such a convergence which have been brought about in a phenotypic characteristics, they are called lysogenic convergence or phage convergence, which have been seen in a prophage. Now, if you will take into consideration different types of bacteria, among them example is Vibrio cholerae, which is the causative agent of cholera, which has been seen in the domestic pest, which has been seen, which is the certain type of disease condition which has been observed during the specific time period or during the specific seasons. Another organism, its name is Clostridium botulinum. Which is again the pathogenic organism. Now, when you will take into consideration this Vibrio cholerae and Clostridium botulinum, in that bacterium, when the prophage is present, then that specific number of organisms in whom prophage is there, they are comparatively more vulnerable. In case of Vibrio cholerae and Clostridium botulinum, where the prophage is absent, in them the virulent activity is comparatively up to lesser extent. Virulent activity is comparatively up to lesser extent because of absence of prophage. Now that means here the phage, that is black phage, who is having the capacity to integrate its genetic material in the host cell. That host cell is either Vibrio cholerae or Clostridium botulinum. In that host cell, the specific genes which are associated with the pathogenicity, that genes are called virulence determining genes, that genes are called toxin producing genes. Now in case of these two organisms, Vibrio cholerae and Clostridium botulinum. Clostridium botulinum is that organism in whom the toxin is produced. Potent exotoxin is produced, and because of production of that potent exotoxin, the abnormalities are created in the individuals in whom Clostridium botulinum gets entered. In case of Vibrio cholerae, the virulence or pathogenicity of the Vibrio cholerae, virulence determining gene of the Vibrio cholerae has been associated with the prophage. So here, due to these reasons, these organisms, when present in the prophage state, they are virulent or they are pathogenic to human beings and animals. In case of the reduction of virulence, a specific induction or excision of the viral genetic material will be carried out from the temperate phage. And when that virulent determining gene or toxin producing gene has been removed, that means prophage has been removed from the Vibrio cholerae and the another organism, that is Clostridium botulinum, the non-pathogenic stains are created, and here the temperate phage will be induced, and from that temperate phage, instead of undergoing the lysogeny cycle, the lytic cycle will be operated, and that has been shown here on this slide in the form of diagrammatic representations. Here, what happens? Please try to pay attention. In case of lytic cycle as well as lysogenic cycle, the first step, that is adsorption or attachment, is common. Second step, that is called penetration, is also common. Once the genetic material is penetrated, here in the lysogenic cycle, that is the temperate phage, what happens? The viral genetic material will be incorporated in a bacterial genetic material. The prophage will be formed. This prophage multiplies. From that, the two prophages are formed. Again, second generation, third generation, up to 20th generation, n number of cells are produced. They are called prophage. In a 21st generation, amongst millions of cells, in any one of the cells, what happens? The genetic material, that is viral genetic material, gets detached from the bacterial genetic material. Under stressful conditions, prophage DNA is excised from the bacterial chromosome. And when prophage DNA has been excised, or when the viral genetic material has been excised from the bacterial chromosome, then later on, the lytic cycle of replication has been continued or has been forwarded. This type of this is called transduction. These types of transductions have been seen in the lysogenic viruses at some stages. There are two different types of transductions seen in the lysogenic patches. They are specialized transductions. The second one is abortive transductions. Abortive transductions are those transductions seen from first generation to 20th generation. From first generation to 20th generation, normally the lysogeny has been forwarded, and in 21st generation, amongst millions of cells, in one of the cell, abortive process has been carried out, and that process is called abortive transduction. The lytic cycle will be operated. Now, this is all about the lysogenic cycle of replication. Have you understood? Any doubt? Now, the next part is cultivation of viruses. Now, see, on the basis of host, viruses have been categorized into how many types? How many types? On the basis of host, viruses have been categorized into how many types? 4 types. Thank you. 4 types. They are: number 1, animal viruses; second one is plant viruses; third one is bacterial viruses; and fourth one is human viruses. Now, listen. Among these things, I have given the example of the viruses which have been transferred from animals to human beings. The host has been changed by the viral. The examples of the diseases in which the virals are positive agents and they have been shifted from animals to human beings. In the last lecture, in case of these 4 different types of viruses, the cultivation, the ways of cultivations are slightly different for the plant viruses, animal viruses, human viruses, and the bacteriophages. Now, among these things, first I will try to tell you about the cultivation of bacteriophage. Then I will try to explain you about cultivation of plant viruses. Then I will try to explain about cultivation of animal viruses, and then lastly I will try to explain about cultivation of human viruses. Now, in case of the first, that is cultivation of bacteriophage. Here I already told bacteriophages are those viruses who have the capacity to infect bacteria. Now, here in this case, for isolation of bacteria, what is done for isolation of bacteria? Normally, in the laboratory, the culture media have been used. For isolation of bacteria in the laboratory, the culture media have been used. Now, is it possible to cultivate bacteriophages in the laboratory using same culture media? Yes or no? Is it possible to isolate the bacteriophages in the laboratory using culture media? Can anyone tell? Yes or no? It is not possible to isolate the bacteriophages in the lab using culture media. Are you agreed with this? Your sentence? No. It is possible to cultivate bacteriophages in the laboratory using bacterial cell and culture media. See, I used the word. It is possible to isolate bacteriophages in the laboratory using bacterial cells and culture medium. Now, you know the association of bacterial cell and culture medium because you have isolated different types of bacteria in the laboratory on the culture media, right? That culture media are available in the form of solid media. Now, these solid media are sterilized and aliquots of these solid sterile medium in a molten form is poured to the sterile Petri plates. On that sterile Petri plates, on that Petri plates to which the culture medium has been poured. Allow that medium to solidify. After solidification, here your aim is to overlap the bacterial cell suspension on that culture medium. For that purpose, use micropipette. 100 microliters or 200 microliters of the suspension will be added to the basal medium plate, followed by using the glass spreader. Spread the suspension all over the plate in a uniform way. This is one technique. Another method is basal medium which is prepared and it is added in a quantity approximately 15 ml per plate. Allow that medium to solidify. After solidification, take a sterile molten soft agar in a quantity approximately 5 ml. Sterile molten soft agar का test tube in which 5 ml soft agar is there. That is taken. To that add aliquots of suspension. Here suspension added to this test tube, soft agar test tube is either 0.5 ml or 1 ml. And that entire content from the test tube will be added to the surface of the basal medium. Allow uniform spreading of that soft agar all over the plate. And for that, what you have to do, you have to move the plate like this, so that that soft agar will be entirely spread. Allow that soft agar to solidify. Now here, just pay attention. Here on the medium cup plate, you have overlapped the specific organism. Consider Escherichia. The medium used is either nutrient agar or Meconium agar. That Meconium agar or nutrient agar will be overlapped with the equalized suspension. Right? Allow it to solidify. After solidification, what you have to do? Here, what is done? The suspension is sprayed. Sewage, which is domestic sewage. This domestic sewage is called the best source of polyphage. Now that domestic sewage, when you are doing the preparation of medium during that time or before that time, what you have to do? You have to treat domestic sewage with the chloroform. You have to treat domestic sewage with other organic solvents. You have to carry out centrifugation of that domestic sewage and consider 5,000 to 10,000 RPM for 20 to 30 minutes. After centrifugation, what will happen? The sewage may, the organic impurities, inorganic impurities, dust particles and other constituents, that is fecal matter or other things, what they will do? They will settle at the bottom. Supernatant may supernatant mean the bacterial cell suspension is there, but that bacterial cell suspension is treated with the chloroform and other organic compounds. So what will happen? The cell lysis will be carried out, and after cell lysis, from that the phages will be released in the surrounding environment. So here the supernatant predominantly contains the phages. Now here again he will say कि bacterial cell suspension जिसमें phage है, what that bacteria, where that bacterial cell will go. Bacterial cell का lysis will be carried out, and that cell will settle in the form of cell debris to the water. Supernatant will be collected. That supernatant in a quantity 0.1 to 0.5 ml, or consider 0.5 to 1 ml of the supernatant is collected and it is added to the phage broth. Now what is phage broth? Phage broth contains specific two ingredients. They are supportive for the phages. Now when it will be added to the phage broth, the phages ka viability will be maintained in a phage broth. Now same aliquots of phage broth, please try to pay attention. Some aliquots of phage broth, that is again 100 microliter or 200 microliters of the phage broth, will be overlaid on the plate which is already overlaid with the cell suspension. Now using the glass spreader, that phage broth which is added will be uniformly sprayed. Now when it will be uniformly sprayed, the phages will be scattered. Phages will be scattered at different roots, at different places. Pehle you have scattered bacterial cells at different places. Then later on you have scattered the phages at different places. Now here the phage which is scattered at this side, the phage which is scattered at this side, the phage which is scattered at this side as well as this side, if finds nearby to him the host cell, then on that host cell this phage will grow. See, during incubation the bacterial cell also tries to grow, the phages will also try to grow. The bacterial cell ka growth will be carried out, and that bacterial cell ka lysis will be carried out by the phage. Now when the lysis will be carried out, you will see the plaque formation, PLAQUE, plaque formation, or here plaque formation. Here you will see plaque formation. Now when you will get the plaque formation after 37, after 24 to 48 hours, incubation at 37 degrees Celsius, when you will observe the plaque formation, you will come to the conclusion that phages have been isolated. Polyphages have been isolated. This is the isolation and cultivation of polyphage or bacteriophages. Next one, isolation and cultivation of the plant viruses. To carry out isolation and cultivation of plant viruses, here is time to pay attention. The plant cells. Cell is the basic and fundamental unit of the living entity. Plant bears plant cells. In the plants, leaves of the plants, stem of the plant, roots of the plant, flowers of the plant, fruits of the plant. The cells are there. Now among this day, one of the component considered plant, leaf. Leaf, a piece of leaf has been taken that has been subjected to the action of different enzymes. Trypsin, chymotrypsin, proteolytic digestions are carried out and from that the single cell line from the leaf of the plant has been obtained. Now that single cell line, when you will culture on the medium, that medias are called tissue culture media. For plant tissue cultures, different types of tissue culture medias are there. See, I am not teaching you the tissue culture, plant tissue culture. I will not give you more details about the medias. Basic part you try to understand. Plant tissue cultures medias are there. Using that plant tissue culture medias, the single cell of the plant cells are cultured in the laboratory, followed by incubation at specific time and temperature. After incubation, what will happen? The single cells, which are the plant cells, that single cells will grow in the specific containers. Now that growth is like this. These are called monolayers. Cells will grow in the form of single layer. That is called monolayer cell line. The monolayer cell lines of the plant cells are prepared. And on this monolayer cell line, the plant virions are allowed to infect plant viruses. Viral suspensions are added in a specific quantity. Aliquots of plant virus suspensions are added to the monolayer. Now, here in this monolayer, what will happen? Cells will grow, viruses, plant viruses will grow in the form of POCK, POC formation. When in the medium, the POCs are formed like this. It indicates the plant viruses have been cultivated. It indicates the plant viruses have been cultivated. Is that clear to you? Now, in case of third type of viruses, that is called animal viruses. The cultivation of animal viruses is a little bit different than the bacteriophages. It is a little bit different than the plant viruses. Now, to carry out cultivation of animal viruses, three different methods have been used, and that three methods are: number one, embryonated chicken egg, or chick embryo method, embryonated egg or chick embryo method, fertile chick embryo method. Second one is tissue culture method. Second one is tissue culture method or cell culture method. And the next one is use of animals, that is called animal inoculations. Now here I will try to explain you the animal inoculation first. Now, in case of three methods, that is animal inoculation, fertile chick embryo method, and tissue culture method, The first one is animal inoculation method. Now, in case of animal inoculation methods, there are different laboratory animals. That laboratory animals are mice, guinea pigs, hamster, rabbits, and primates. I am repeating again: mice, guinea pigs, hamster, rabbits, and primates. These animals are used for doing the inoculations. Now, in case of animal inoculation, the first and the most important point is selection of animals. Now, to select the animals, normally what is done? The healthy animals which are free from the diseases, they have been preferred. Now, most of the diseases which are transmitted through the community, they are called different types of communicable diseases. There are some type of non-communicable diseases also. Now, those animals which we will select for the cultivation of viruses, either guinea pigs, mice, hamsters and rabbits, they should be free from, or they should be healthy, they should be free from different types of communicable diseases. Please try to pay attention. In case of vaccine manufacture, in case of serum manufacture, normally little bit larger animals like horses are used. That horses are having the capacity to infect one particular infecting organism, that is called actinobacillus virus. The horses should be free from such a communicable disease. In a similar way, the mice which are used, guinea pigs or rabbits which are used, they should be free from different types of communicable diseases. They should be healthy. In addition, another important thing is suckling mice lesser than 48 hours old. They are also commonly used for the animal inoculation studies. Suckling mice are susceptible to the toga virus and coxsackie virus, which are inoculated in intracerebral and which are inoculated by intracerebral and intranasal routes to these suckling mice. Now, in case of viruses, most of the viruses have been inoculated by intraperitoneal route, or they have been inoculated by subcutaneous route. Now, when after inoculations, please try to pay attention. After inoculations, that animals have been kept separately. They are kept away from the uninoculated animals. They are kept in separate cages. They have been provided with the food, balanced food. They have been provided with the balanced food. They have been examined during the incubations. After inoculations, then they have been incubated, and during the incubations they have been examined. Now, here, intraperitoneal and subcutaneous route. When the virus has been inoculated, then the virus will multiply either intraperitoneally or subcutaneously. After specific population density of the virion will be reached in the inoculated animal. A specific type of pox have been developed on around the site of subcutaneous or intraperitoneal inoculations, around the site of subcutaneous or intraperitoneal inoculations. When the virus population has been reached up to sufficient level, then the animal to which is inoculated with the virion, that animal shows specific signs and symptoms of the disease. Then the killing of that animal will be carried out. The killing of that animal will be sacrificed. After killing or sacrification of that animal, a specific organ in which the virus has been multiplied, that organ has been separated. It will be subjected to the action of different enzymes, proteolytic enzymes. Trypsinization will be carried out. The viruses have been separated, and the cell debris have been separated. In this way, from the animal inoculation, using the animals, the cultivation of viruses has been carried out. Now, when we take into consideration, live inoculations was first used on a human volunteers for study of yellow fever virus. In case of animal inoculations, diagnosis, pathogenesis and clinical symptoms are found to be very well determined. In case of animal inoculations, the immune system of the animal will be activated, antibodies are produced. In case of animal inoculations, primary isolation of certain viruses will be possible. Mice is the small laboratory animal which will provide best model for studies of viral replications. Studies of immune responses, epidemiology and the viral replications, that is called oncogenesis. That studies are also carried out simultaneously using the animal inoculations. If you will take into consideration, as compared to the other inoculations, this animal inoculation is little bit expensive. Difficulties will arise in the maintenance of animals. Difficulty is also there in selection and choice of the animals for particular virus. Some viruses cannot multiply in the animals. They cannot multiply or they cannot grow in the animals. Some viruses cannot show the specific signs and symptoms in the animals for particular diseases. When you take into consideration mice, which is a smaller animal, it will not be useful for the vaccine development. In case of animals, the production of mutants is also little bit increased, and if the mutant will be escaped, then it will lead to the generation and escape of mutants, and if the mutant will be escaped, then certain type of epidemic or pandemic conditions will be created in the world or in the society. In case of animal inoculations, other things which are related to the animal welfare systems, issues are there with the animal welfare system. They will not allow you to use the animals for the cultivation of viruses. So here, second method is found. Second method has been taken into consideration in the year 1931. whose Pasteur was the first scientist who had used the embryonated egg for cultivation of animal viruses. He had used the embryonated egg for cultivation of animal viruses. Now, when you will take into consideration embryonated egg, to whom we will call embryonated egg? Just pay attention. The eggs, which are normal eggs, they are not poultry eggs. Please try to pay attention. These eggs are allowed to incubate along with the hen. See, because of feeders of the hen, the specific conditions have been created, and when these eggs are incubated and along with the feeders of the hen, for consider 7 to 8 days time period. The eggs which are normal, in that egg, the embryo will be developed in these 7 to 8 days. Now, such eggs which are incubated with the hen for 7 to 8 days below feeders, in them the embryo will be developed. That embryo is used for cultivation of the viruses. In addition to that embryo, there are few other sites from the fertile chicken egg. Now you all have seen the egg, which are poultry eggs. Normally, have you seen the chicken eggs normally? Yes, no? So that egg contains outer shell. That shell is made up of calcium. Below that shell, there is a choriolantic membrane, which is too thinner. Below that shell, there is a choriolantic membrane. Below that choriolantic membrane, there is a odontic cavity and odontic fluid. Below that, again, at one place, amniotic fluid and amniotic cavity is there. In the egg, yolk sac is there. In the egg, embryo is there. Now these are different sites. See, I told you, choriolantic membrane, odontic cavity or odontic fluid, then amniotic cavity or amniotic fluid, yolk sac and embryo. These are 5 different sites. In case of cultivation, for cultivation of viruses, all these 5 sites are useful. When you will take into consideration choriolantic membrane. Choriolantic membrane is useful for cultivation of smallpox virus. Odontic cavity is useful for cultivation of influenza virus. There are several advantages of chick embryo method, as they are packed in their cells, and these chick embryos are having the natural resistance against different types of bacterial contaminants. This chick embryo is easy to handle. Fertile chicken egg is easy to handle, and due to that, these fertile chicken eggs are having used for cultivation of different types of viruses. When you will take into consideration choriolantic membrane, there are different types of viruses. They are mainly herpes simplex virus, pox virus, Raux sarcoma virus. They have been inoculated in the choriolantic membrane. In case of amniotic inoculations, measles, mumps virus and influenza virus. Mumps virus and influenza virus. In case of yolk sac inoculations, in case of yolk sac is used as a medium for cultivation of herpes simplex virus. In case of amniotic inoculations, influenza virus, mumps virus, Newcastle disease, and avian adenovirus. These are few viruses which have been cultivated in the amniotic cavity or amniotic fluids. Now, when you will take into consideration the chick embryo method as compared to the animal inoculations, this chick embryo method is less expensive. Isolation and growth of the virus is comparatively wide, up to wider extent. Ideal substrate, chick embryo will be used as the ideal substrate for viral replications. Chick embryo will be used as the ideal substrate for cultivation of viruses or obtaining the growth of the viruses. Different types of mammalian viruses have been also cultivated using the chick embryo technique. This method is cost effective. Sterile and wide range of tissues and fluids have been used in the chick embryo method. Embryonated eggs are readily available on the earth. Different types of, they are free from contaminating bacteria. In addition, different types of latent viruses, from them also this chick embryo method is little bit free. Defense, some sort of defense has been involved in the isolation and cultivation of viruses. That defense is specific or non-specific defense for the embryonated egg is there up to lesser extent, not up to more extent. When you will take into consideration, it is traditionally used or widely used method for the vaccine production. The size of inoculum is comparatively lesser. There are different types of viruses. There are different sizes of the viruses for the multiplication. So one egg and different size will not be possible. It will create the problems in the purification, during the purification. This is second method of inoculation. Now the third method of inoculation is using the cell cultures or using the tissue cultures. Now this is cell culture and tissue culture. There are different types of cell cultures and tissue cultures. Little bit 20 minutes time will be required for explanation of that. You have next lecture Indre madam. ठीक है